These 9 do not kill their hosts immediately. They have better uses for them. The jewel wasp does not paralyze its prey. The female stings a cockroach twice, first in the thorax to temporarily disable the front legs, then precisely into the subesophageal ganglion, the specific nerve cluster governing the escape response, injecting venom that selectively blocks dopamine receptors and removes the cockroach's ability to initiate its own movement. The cockroach remains fully conscious and capable of moving if prodded externally, but will not walk on its own. The wasp clips the antennae, drinks hemolymph from the cut ends, and leads the cockroach by the shortened antenna into a prepared burrow. The cockroach follows without resistance. The wasp lays a single egg on its abdomen and seals the burrow. The larva feeds on the still-living cockroach over several days, consuming non-essential organs first to keep the host alive and fresh for as long as possible. Ophiocordyceps unilateralis infects carpenter ants through contact with spores on the forest floor. After infection, the fungus infiltrates muscle fibers throughout the ant's body, not brain tissue, and produces compounds that manipulate the muscle cells directly. Infected ants leave their colony, climb vegetation, and bite onto a leaf vein at a height that precisely matches the temperature and humidity required for fungal sporulation, between 25 and 26 degrees Celsius, typically 25 to 30 centimeters above the soil. This height selection is consistent across individual ants and across different forest sites. The ant dies locked in position. A fruiting body grows through the back of the head and releases spores downward onto the foraging trail below. Healthy colony members recognize infected individuals early and carry them away from the nest before they die. Sacculina is a barnacle. Its larval form is a tiny free-swimming organism that locates a crab, finds a joint in its exoskeleton, injects a packet of its own cells into the crab's body cavity, and discards its outer shell, every visible part of it, permanently. The injected cells grow through the crab's internal organs as a branching root system over weeks, eventually producing a bulge on the underside of the crab's abdomen, Sacculina's reproductive sac. The infected crab stops molting, stops regenerating lost limbs, and is permanently castrated. If the host is male, Sacculina produces hormones that feminize its body shape and behavior, because female crabs perform specific brooding behaviors that the barnacle requires. The crab grooms and aerates Sacculina's egg sac as if it were its own offspring, for the rest of its life. It will never reproduce. Glyptapanteles, a parasitoid wasp, lays several dozen eggs inside a caterpillar host. The larvae develop inside the caterpillar for days, feeding on hemolymph while avoiding vital organs. They then synchronously chew through the caterpillar's body wall and spin cocoons on the outside of its body, dozens of silken cases on the surface of a still living host. A small number of larvae do not emerge, they remain inside. These remaining individuals are responsible for an unusual outcome. The caterpillar stops feeding and moving, anchors itself to its perch, and begins thrashing violently whenever any predator approaches the external cocoons. It defends them actively until the adults emerge. Caterpillars that were experimentally manipulated to prevent this defense behavior produced fewer surviving adult wasps than those that performed it. The caterpillar does not survive. Leucochloridium paradoxum is a flatworm that requires a bird and a snail to complete its life cycle. Inside the snail, it forms brood sacs in the eye stalks, pulsating, brightly colored structures that expand and contract rhythmically inside the transparent eye stalk tissue, stretching the eye stalks to several times their normal length. The pulsation rate and coloration vary across different worm species to match the visual search image of the specific bird species present in that region. Birds peck off the eye stalk, consuming the brood sac, which is shed in the bird's feces and eaten by another snail. The snail's eye stalk regrows. The infection recurs. Hairworms, Nematomorpha, spend their larval stage growing inside terrestrial insects, crickets, grasshoppers, mantises, absorbing nutrients through their skin over months. When mature, hairworms can be longer than their host. They then produce neuropeptides that mimic the host's own signaling molecules, manipulating its behavior. The infected host seeks water and jumps in. Once submerged, the worm exits through any available opening, sometimes through the abdominal wall, and begins its aquatic adult reproductive phase. The host drowns. In stream ecosystems where hairworms are common, the quantity of insect biomass entering the water from this mechanism has been measured as ecologically significant. The parasite drives a detectable transfer of organic material from terrestrial to aquatic food webs. Toxoplasma gondii can only sexually reproduce inside a cat's intestinal lining. In rodent intermediate hosts, it forms cysts throughout the body including in brain tissue. Studies of infected rats show reduced or reversed fear responses specifically to cat urine, a stimulus that normally triggers a strong innate avoidance response in uninfected individuals. Infected rats approach rather than avoid cat-scented areas in controlled experiments. The suppression appears to be targeted, it does not extend equally to other predator odors. Approximately 1/3 of the global human population carries latent Toxoplasma infection. Chronic infection in humans has been associated with statistically elevated rates of traffic accidents, altered reaction times, and modified personality profiles in large-scale studies, though causation at the individual level has not been established. The lancet liver fluke Dicrocelium dendriticum requires 3 hosts to complete its cycle: a land snail, an ant, and a grazing mammal. After passing through the snail and being expelled in slime balls consumed by ants, the larval flukes infect an ant colony. One larva, and only one, migrates to the ant's subesophageal ganglion. This individual does not reproduce, its sole function is behavioral manipulation. At cool temperatures, when grazing animals are active, infected ants climb to the tips of grass blades and remain there motionless until the temperature rises. In the morning warmth, they descend and resume normal behavior. The single sacrifice larva survives if the ant is not consumed by a grazing animal, and continues manipulating the host for another evening. The remaining larvae inside the same ant are carried into the mammal host when the ant is eaten. Hymenoepimecis argyraphaga, a Costa Rican parasitoid wasp, lays its egg on the abdomen of a living orb-weaver spider. The larva feeds on the spider's fluid reserves for days while the spider continues building its normal orb web. Approximately 1 week before pupation, the larva injects compounds into the spider that cause it to abandon its web design permanently. The spider instead builds the same simplified structure repeatedly, parallel zigzag threads with a reinforced central hub, a structure never built under normal circumstances. This modified web is structurally superior for suspending the wasp's pupal cocoon against weather and vibration. When the web is complete, the spider stops moving. The larva consumes it, then hangs its own cocoon from the modified structure the spider built. The spider's final construction project was its own replacement. 9 parasites. 9 hosts that were more useful alive than dead, for a while. Tomorrow's fact involves an ability scientists still can't fully explain. This channel posts a new animal fact video every day, subscribe so you don't miss the next one.